Okay, so Adrian, are we live? Are we recording? Wala pa. Ayan, okay na? Napaka-active ng ano ko. Sir Marco, napaka-active ng ating moderator, no? Ay, ayan, oh, di ba? Very active talaga siya. He's very dependable, actually. Oo. Pwede ko bang i-hire yan? Pwede ko bang i-hire yan as a moderator for my for my lectures tomorrow? For my lectures tomorrow. I know for my blood bank lectures. Charing. Anyway, may blood bank pa ako sa Sunday eh. I-hire ko si Adrian. Charing. Charing lang. But anyway, kung may time naman siya, wala lang, charing lang. Char lang. Alright. Kawawa naman si Adrian. Dami na nga inuutos si Sir Mac. Alright. Let's move on. Common diseases associated with Escherichia coli. Basically, you guys already know this because anything that's Escherichia coli is basically is normally found in human excrement or feces or poop, and any place where it doesn't belong to will definitely cause either of the following: UTI, sepsis, meningitis in infants, diarrhea. Okay, diarrhea. I thought, sir, it doesn't, it's found as normal flora in the stool. Well, you'd be happy to know that there are other serotypes of, um, I don't think you would be happy, but you'll be happy to know that that there are some species of, rather strains of Escherichia coli that actually causes diarrhea, um, and we're going to talk about them later on. Right. So meningitis in infants is associated to E. coli. Uh, in E. coli may be correlated with what Landsfeld group of strep also. There's there's a correlation between E. coli and the Landsfeld group B. Okay. So there are some theories uh, that the Streptococcus group B actually allows the passage of E. coli, or has some sort of mechanism which allows the passage of E. coli in the blood-brain barrier of infants. And that hasn't been proven yet, but there has been correlations with that one. And a lot of studies have actually proven this. If you actually took the time to read your notes, or rather your books, you will see that there is a table there. I'm not sure if it's on the group B streptococcus discussion or in E. coli, but there is a study there that has, I think NCBI or Medscape also has a study about the correlation between E. coli and Strep B. And yeah, let's move on. That's the reason why Strep B is also screened in infant in antenatal screening for pregnant women. Yeah, antenatal nga eh, kaya for women before they deliver. Okay, that's the reason why it's important for us as microbiology techs to identify group B in vaginal swabs of women, of people, of women who are about to give birth. Now, let's go with the tests. IMVIC results. IMVIC results. I don't want to stress this enough. I want to stress this enough that this is important in the board exam. IMVIC results basically indole, methyl red, Voges Proskauer, citrate. All right. So IMVIC result of your E. coli is positive, positive, negative, negative. TSI is acid, acid. Sometimes there's gas formation and sometimes there's not. But the most important thing that you need to remember is the IMVIC and the TSI result. It's positive, positive, negative, negative. I think up till now, even though I'm not using biochemical testing anymore, I can still remember that E. coli will produce a positive, positive, negative, negative result. And you have to remember that. There will be a lot of questions in the board exam and even in your exams in general here in college, or as undergraduates, to that will ask you about these particular tests. So please take them into heart. Even also the what do you call this, the Klebsiella species. But we're going to talk about the five strains of E. coli, or five serotypes of E. coli. We have enterotoxicogenic E. coli, enteroinvasive E. coli, enteropathogenic E. coli, enterohemorrhagic E. coli, and enteroaggregative E. coli. Now, among the five types of E. coli, which is considered to be non-invasive and cannot produce a toxin, it is actually enteropathogenic E. coli. The most common pathology associated with this one is what you call infantile diarrhea. All right, infantile diarrhea. And it often appears in the patient's stool, in watery and mucoid stools. Okay, and we use different types of culture media for that one. But for infants, we're not supposed to see E. coli's, not infants, newborn. We're not supposed to see E. coli yet because what they're eating is they haven't been colonized yet, especially premature babies. You're not supposed to see any growth of E. coli in their stools. Okay, and I think there's a book there that says that after 10 days, that's the time they're going to develop their own bacterial colony. But yeah, we're not supposed to see any E. coli. And if there is E. coli, it is considered to be a nosocomial infection. Probably the hand hygiene of the healthcare professionals, or even the healthcare professionals, which includes nurses and doctors, or even the non-healthcare professionals, are not properly washing their hands in the neonatal ICU. So this is the most common type of E. coli that you would see in the neonatal ICU. Okay, it's associated with nosocomial infantile diarrhea. Okay, enteropathogenic E. coli. Now that enteropathogenic E. coli is out of the way, we're going to talk about the toxins released by ETEC. What is ETEC? Enterotoxicogenic E. coli. Let's talk about enterotoxicogenic E. coli now. There are two types. We have the heat labile and heat stable toxin. And the heat labile one closely resembles the bacterial toxin, the cholera toxin. Okay? The heat stable one increases net fluid secretion in the intestines by stimulating guanylate cyclase. What is guanylate cyclase and what is it? Basically, guanylate cyclase is a biochemical interaction that happens in the large intestine. It allows the body to absorb water from the food that we eat in the large intestine. And if there's any toxins that if there's a toxin in the stool, if there's a toxin in a patient's stool, and uh, it interferes with and it stimulates guanylate cyclase. What it does is it binds to the receptors in the large intestinal epithelium, and it stops the it stops the absorption, and it increases the fluid secretion in the large intestine. So it is supposed to be it's going to absorb the waters in what the H2O molecules in the stool. However, because guanylate cyclase is activated by this heat stable toxin, it actually does the opposite. And hence it causes the patient to become dehydrated. So you might see this in cases, for example, you might see it in case presentation. 55-year-old male who traveled from blah 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 blah, suffering from suffering from fever, fatigue, and clear signs of dehydration. Ay, oh, dehydration. This might be associated with E. coli, but I need to know what first. That they're going to talk about certain tests, and you need to remember that E. coli is actually one of those organisms that you need to take into consideration. Okay? Now, sorry, yung clock lumabas. What is the disease associated with ETEC, or enterotoxicogenic E. coli? That's the reason why I need you guys to be familiarized with the patient's history as well. Because if in the case presentation they will tell you that the patient has traveled, you will associate it with the following terminologies. the following diseases. We have traveler's diarrhea, Montezuma's revenge, and the Turista's disease. Okay, because this is the type of this is the type of E. coli that you might get from water in places like Mexico, places like uncle. I think it's pretty common in South Africa. Okay, and then it's also present in some places like Myanmar and in places around India. All right, wherever there's contaminated water. Anyway, let's move on to the Shigella-like E. coli. The Shigella-like E. coli is what you call the enteroinvasive E. coli. Okay, and aside from Shigella, aside from Shigella, this is the only organism that can cause. This is one of the organisms rather that can cause dysentery. Now, what's the difference between diarrhea and dysentery? Anybody? Anybody in the chat can tell me? Yeah, I'm sorry, Adrian. I'm sorry, Adrian, if I just realized the microphone is away from my mouth. But yeah, um, so what are the types of E. coli? What's the difference between diarrhea and dysentery? Somebody raised their hand. I didn't see. Who was that? Unmute yourself and tell me the difference. Wonderful. Okay, so she's partially correct. So I will accept her answer. Joanna's answer was a little bit incomplete. Um, Dysentery is actually characterized by presence of blood and mucus, which are cardinal signs of, which is basically one of the signs of an inflammatory disorder. In diarrhea, this is only watery stools, characterized by watery stools. There may be some blood, there may not be some blood, depending on how often the patient defecates. Okay, so in diarrhea it's more of the toxicogenic activity of a particular organism, while in dysentery it's more of the invasive characteristic of the organism. Okay, so please put that into your noggins, let it marinate because it might be useful for you in the exams. Now, enteroinvasive, as I mentioned before, can cause Shigella-like infection. All right, and we're going to go on with EHEC, um, because EHEC has a special or EHEC enterohemorrhagic E. coli can actually cause hemorrhages in the large intestine, and this is because of the one specific toxin, which is the virotoxin. The name is derived from the viro cells, which is derived from African green monkey cells. Just for your information, I don't know if it's going to be asked in the board exam, but I want you guys to remember virotoxin, viro cells, African green monkeys. All right. Now, similar cytotoxin is produced by by Shigella dysenteriae cytotoxin. Sir, akala ko ba kanina sinabi mo enteroinvasive yung Shigella-like? Shigella-like lang siya. Why? Because it is invasive. Okay? It causes dysentery because it is invasive. Remember, paano ba tayo nagkakaroon ng inflammation? Kailangan ma-expose tayo sa foreign material or foreign antigens. And in this case, because the E. coli is becoming invasive, it produces Shigella-like symptoms or infection. But in the case of enterohemorrhagic E. coli, it causes it mimics rather the cytotoxins produced by Shigella dysenteriae. All right, so that's the difference, huh? Other one acts like the other one acts like Shigella dysenteriae. The other one produces toxins similar or biochemically similar to that of Shigella dysenteriae. So please don't forget the difference, enterohemorrhagic and enteroinvasive. Okay? Now, it's important to note also, sorry, kumain ng ano. Sorry. Sorry, how very rude. All right. So, it's important to note also the difference the zero group or serotype of EHEC because it's going to be asked in the board exam most probably. And one of the things that you need to remember is the serotype H70157. It can actually be detected with SMAC or sorbitol McConkey. The colonies will appear colorless, and the only E. coli serogroup that is non-sorbitol fermenter. All right, so all serogroups of E. coli are sorbitol fermenters except for H7057. Oy guys, ano nga itong H at saka O na to? What are these? These are antigens. What does H stand for again? Review question from 30 minutes ago. What is H? Anybody? What is this antigen for? H antigen, O antigen. Ang daming antigen na kailangang alalahanin. Sino? Sino doon? Age? Ah, okay, H. H pala ang tinutukoy niya, sorry. Hindi ko narinig ng tama kasi there's the sound at the back. What about the O antigen? What's the O antigen going to take into what's the O antigen for? All right, wonderful. And that is located where? Wonderful, wonderful. All right. Now let's move on to the diseases associated with the enterohemorrhagic E. coli. So there are basically two types of diseases that you might be familiar with. I don't know if it was discussed to you guys, but hopefully it was. So this will just serve as a recall. Okay? Hemorrhagic colitis and the hemolytic uremic syndrome. All right? Hemolytic uremic syndrome, it is going to be a question. If it's not going to be asked in the microbiology exams, it's going to be asked in your hematology exams because which of the following which of the following would cause hemolytic uremic syndrome? It will give you a couple of choices, and then at the very bottom it will say E. coli infection. E. coli variant serogroups of E. coli. That was supposed to be your answer. Okay, it will give you a lot of random answers, and then one of those choices would say enterohemorrhagic E. coli, and I assure you that is the correct answer. Okay, I've encountered it several times in the question banks whilst I was reviewing for the board exam, and my friends and I, and even my previous students, have also can also testify that this is actually going to appear in questions such as these. All right. Especially, I think in hemolytic when the discussion talks about hemolytic anemias. All right. So those are the two diseases. And what is the most severe manifestation or severe disease state? The hemolytic uremic syndrome. Why? Because um, the uremia causes the cells to lyse, as well as it can go the urea the urea can be split into ammonia, and it can cause a wide variety of organ damage. What do I say? Wide variety of organ damage. It can damage the liver because the liver will compensate for will will need to neutralize it into forming a no ammonia molecule. Will need to neutralize the ammonia molecules in order for it to be released in the body. So it can kill the liver. Um, Urea in the kidneys can also be affected, thus forming damage in your thus damaging the kidney cells or the nephrons. It can also go to the brain, resulting to Reye's syndrome. So a lot of things can happen when a patient suffers from hemolytic uremic syndrome. And that's the reason why blood cultures are always taken into account whenever somebody is sent to the ICU, because many patients die from hemolytic uremic syndrome. Many septic patients die from hemolytic uremic syndrome, especially when they are colonized by E. coli. All right. So there was one case, I think, when I was in the Philippines. They admitted the patient in the ICU 3 days prior to him suffering from the onset of high-grade fever and bacterial infection. Anything, the labs are clearly pointing out into bacteremia, but the patient didn't want to go to and get admitted. So after the third day, he finally decided to get himself treatment, and then get admitted to the hospital, and was directly sent to the ICU. Blood culture shown after 48 hours indicate that there is severe gram-negative septicemia. And what grew was actually Escherichia coli, and it was too late for the patient, unfortunately, because he had multiple organ failure, and the final diagnosis in his medical records before prior to his demise is hemolytic uremic syndrome. Okay, so this is one of the reasons why I still remember HUS up till to this day, because I remember clearly the pathologist explaining to us what is hemolytic uremic syndrome. All right. So now, this is the type of E. coli where pathogenic mechanism is still unknown. So we still don't know up till now. I actually reviewed this enteroaggregative E. coli. If somebody will be able to understand if somebody can be able to shed light into it, I will share it in a later video. But up till now, there is still no pathogenic mechanism for enteroaggregative E. coli or AIC. Now, let's move on to the Enterobacter and Klebsiella species. All right. So Enterobacter and Klebsiella are have similar TSI and IMVIC reactions. IMVIC is negative, negative, positive, positive, opposite of that of E. coli. IMVIC is positive, negative, negative, positive, positive. Whilst for E. coli, what is it? Positive, positive, negative, negative. See the difference for methyl red and Voges proscour? It's almost certainly always like this. Okay. Now TSI is A over A plus gas. Motility. Let's check for motility. Klebsiella is non-motile, of course, because there's no a lot of the medically important Klebsiella species don't have any uh, any flagella, whilst Enterobacter is motile. Klebsiella is negative for ornithine decarboxylation, whilst Enterobacter is positive for ornithine decarboxylation. All right, so I discussed them together based on biochemicals, but we need to move on with the other names for Klebsiella pneumoniae. So Klebsiella pneumoniae is also known as Friedlander's bacilli, or the long term, if you want to sound posh, is Bacillus mucosus capsulatus. Okay, Bacillus mucosus capsulatus. The most important pathogenic determinant is the capsule. Oy, so does it cause meningitis? We'll talk about that later. But the appearance of this organism is that it produces mucoid colonies that tend to string. Now, in my uh, less than more than half a decade experience working in the microbiology lab, it's always going to appear mucoid. And to be honest, I'm so tired of seeing Klebsiella that that sometimes I just want to write Klebsiella on the result forms. But unfortunately, we have to do susceptibility testing. So I need to still identify the microbes. So I need to do ID. But when I was there in the Philippines, the person who was training me said to me, this was always will say we'll always have a bet. um, this is Klebsiella. I would often disagree. We would only look at the blood agar plates, and I would always disagree with him. And unfortunately, I was still naive back then. But now that I've been I'm I've been releasing results for a very long time, I have enough experience to say that I can identify them without looking at the biochemicals. But for the sake of complete identification and ruling out mixed cultures, you still need to do identification. Okay, we can't medtech laws. You should not. You should always follow the protocols because it will be under. You will be what you call this considered as as a professional who neglected his or her duties as a medtech. All right. So Klebsiella species that is associated with purulent sinusitis or sinus infections. This is Ozone. All right, Klebsiella ozone. And then Klebsiella species associated with granuloma of the right nasal or the nose and the oropharynx is going to be a very long one for you guys. Klebsiella rhinoscleromatis. All right. Now, how is Klebsiella going to differ from other species of Klebsiella oxytoca going to differentiate from the other ones? You can differentiate it using indole motility. All right, it's going to be differentiated using indole motility. Somebody raise their hand, unmute yourself, and then tell me what's up because I'm going to continue on because we have a lot of things to tackle on. All right, now let's move on to the predominant isolate of Enterobacteriaceae. Most isolates in the laboratory that you're going to encounter is Enterobacter cloacaceae or cloacaceae. Okay, and the yellow pigment of Enterobacter species, the yellow pigment producing Enterobacter species is Enterobacter sakazaki. The pigment intensifies when it's actually incubated at room temperature. Okay. Now I've never experienced this one because unfortunately I wasn't able to isolate this one during my half a decade experience in the lab. But some of my friends who's working there in the Philippines have actually isolated it, and they would testify it to be true. They would testify it to be true. All right, now let's move on to the urease activity. Increased urease activity or a rapid urease production can be seen in the species in Enterobacter gervoviae. Yeah, gerjoviae. Sorry, gerjoviae. So the only Enterobacter species that is a rapid urease producer is Enterobacter gerjoviae. Now, I need to ask a review question. When would you say something is a rapid and when would you say that something is a slow urease producer or urease reactor? Differentiate it for me. Can somebody, it's a review question from from an earlier discussion. How would you differentiate rapid and slow urease activity? I said it earlier. Let's try and ask um who else is here? I've only been hearing Adrian and Mera. Mera, are you there, girl? All right, briefly discuss to me what's the difference between rapid and slow urease activity. Nakalimutan? Oh, she kind of forgot. All right, so I'll try answering for you. Rapid urease activity is you're going to get a positive result 2 to 4 hours. Slow urease activity is you're going to get a result more than 4 hours. Is it important in the laboratory? No, but it is important in differentiating certain species of microorganisms. Okay? So if you're testing if you're testing for a specific microorganism, you need to differentiate the urease activity. Unfortunately, for manual detection, of the organism or manual speciation, biochemical speciation, it would be very difficult, especially with a large load of sample. But for automated biochemical speciation, it would be easier because the machine can run several samples at a time. All right. Now, This is a table that I want you guys to be familiar with. Okay. Now, I don't want you guys to be intimidated, but please be mindful of these results. All right. So how can you differentiate the following? Do I need to remember all of them? Um, I want you guys to compartmentalize the things that you're going to learn. Um, is Klebsiella pneumoniae and Klebsiella ophthitoca. Both of them, all of them, both of them will test positive for LDC, but oxytoca will most definitely produce a negative result to ODC and arginine dehydroxylation. Okay, whilst Klebsiella pneumoniae will produce variable results. This sign means that there is variable results. Okay, next, Enterobacter aerogenes and Enterobacter cloacae, they will produce a reaction like this. Phantomea agglomerans. Why did I put it here? Well, as I mentioned before, they have been classified under under the Enterobacter species. You could call them Enterobacter herbolica, and these are the reactions. The most variable one, Phantomea agglomerans. All right, Enterobacter cloacae, negative for LDC, but positive for the rest. And then Enterobacter aerogenes will be positive for both LDC and ODC, and then ADH negative. All right, try to compartmentalize what you're learning. Don't just memorize the whole don't just memorize the whole table. Okay? Now, let's move on to Serratia. So as I mentioned before, Serratia is going to produce red pigments in the culture plates. This is Serratia marcescens and Serratia rubidea. All right? So the name of the pigment is actually prodigiocene. This is important. Why did I mention it? Because some exams they will ask you which of the following organisms produces prodigiocene. And from the name itself, it doesn't sound like it's producing red pigments. Okay, so please be mindful of this. Okay, and these are the two organisms that we're going to talk about. Serratia marcescens and Serratia rubidea. They're the red pigment producing colonies. And is it true that they produce red colonies? Yes. All the time, they will produce red colonies, even in Makonki agar. All right, notable biochemical characteristics of Serratia marcescens. Please don't be intimidated. Try to compartmentalize what you're going to learn because they are DNase positive, gelatinase positive, and lipase positive. I need you guys to familiarize yourselves with these two tests. There are two organisms that will test positive. There are two organisms that we've discussed that tested positive with DNase. Can you give me one organism that can test positive with DNase from last week's discussion? Anybody? Review. DNase positive organism. Remember, my discussion was that it tested the DNase media is colored green, and when there's DNase production by a particular organism, it will turn the green media into a clear zone. Which organism is that? Okay, so I'm gonna since we're in a time crunch, I want you guys to answer this question for me because this is important. Because sometimes, ay, ang bilis sumagot. Hindi ko pa natatype ang question. I love it. Thank you so much, Mayra. Now, gelatinase and lipase is actually another thing that you need to remember for Serratia marcescens, Serratia species in general. Because they're the only ones that will test positive for these tests. No other organisms, you will not find any other organism in the Enterobacteriaceae family that will test positive for these things. All right. So if in case you see details of a particular organism in a case presentation and they mention gelatinase and lipase positive, automatically don't look at any other or don't look at the other choices. Search for the one that is Serratia. That's uh, that's how they they helped us out in the board exam review centers. All right, so Serratia odorifera produces the following characteristic odor. What particular odor though? The rancid potato-like odor. Is it not? Is it important to remember? No, but I just added it there for you guys. All right, and I think this one is from a review center. If I'm not mistaken, it was from Axe Review Center. So I don't take full credit in this particular slide. It was a it was mentioned to us by I think Miss Analea Navarro, who was our reviewer. All right, now let's move on to how the following are differentiated. Actually, it's not the screening test is gelatinase lipase for Serratia species, but then you have to differentiate them with another set of tests. There's actually a differential test for them. And these are the tests. Ornithine decarboxylase and arabinose fermentation. Oh, arabinose. Please don't forget, it's another sugar. Now this time, I want you guys to remember the lightning bolt, a lightning bolt, a lightning bolt, basically, because this is important for you in remembering it. Lightning bolt. All right. So the only positive results would be for Serratia marcescens. For ornithine decarboxylation is Serratia marcescens, Serratia liquefaciens, and then negative for Rubidea. Arabinose will test positive for both liquefaciens and Rubidea. So this is basically my how we were taught how to remember this. When you're talking about Serratia, MLR, marcescens, liquefaciens, Rubidea, just remember the lightning bolt. Write ODC and arabinose fermentation, and then remember there's a lightning bolt. Why? Because the results resemble a lightning bolt. But again, please don't forget ornithine, decarboxylase, and arabinose fermentation. All right. Now let's talk about the species of. Let's talk about the diseases caused by Serratia species. It causes nosocomial infections, all right, meaning that most of the time they will be isolated in clinical samples, and they don't really appear in general to patients who are in the public or community. It's not a community acquired infection. It's usually a nosocomial infection, and it can cause severe endocarditis, pneumonia, and bacteremia. Actually, we had a patient who had severe bacteremia and had died from the infection, and the organism in question was Serratia marcescens. All right. Now, we're going to move on with another organism or another family of microorganisms, which is Salmonella. Am I going too fast for you guys? It's not like before, right? Warm up lang yung discussion natin last week because tinatry ko pang i-gauge kung paano kayo matuto. So I think a fast-paced discussion would be appropriate when we're discussing when we have a lot of material to discuss. So I tried my best to condense the material for you. Okay. So examples of diseases associated with salmonella is enteritis, systemic infection, and enteric fever. What do you mean by enteric fever? Well, basically um, you're suffering from diarrhea and vomiting, and then you also have fever. All right, so it's associated with a plethora of diseases, but in general, these three are the ones that are characteristic for Salmonella species. Okay. Now, species of Salmonella according to the Kaufman-White scheme. You have to remember the four organisms that we will talk about. We have Salmonella typhi, cholera suis, Salmonella typhimurium, and Salmonella derby. These are the species that can be identified using the Kaufman-White scheme. All right. Kaufman-White scheme: Salmonella typhi, Salmonella cholera suis, Salmonella typhimurium, and Salmonella derby. All right? Now, let's move on to the Ewing classifications. Ano ba yan, sir? Ang dami namang kailangang alalahanin. Huwag kang mag-alala. Huwag kang mag-alala, Adrian. Huwag kang mag-alala. Kasi madali lang siyang maalala. So for Kaufman-White scheme, just remember these two. Well, Kaufman-White and the Ewing classification. Just remember the first two, and then these are the only things that will change in the Ewing classification. So in the Ewing classification, you still have typhi and typhimurium, typhi and cholera suis, but what's different in the Ewing classification is they added group, a third group, and then there's a serotype for each. Now, there is Salmonella typhi cholera suis, and this time they added Salmonella enteritidis. But you can serogroup them into either Salmonella serotyphimurium, a Salmonella enteritidis serotyphimurium, and then Salmonella enteritidis seroderby. All right? Easy peasy? Okay. Now, let's talk about the non-motile salmonella. We have Salmonella polorum and Salmonella gallinarum. Okay? They are the when you identify Salmonella species, you need to identify whether or not they are motile. All right? You need to identify whether or not they are motile. And again, how do we test for motility in manual biochemical testing? Anybody can answer? Wala. Walang sasagot? O sige, magtatawag tayo. Um, Baka nakatulog na kasi sobrang bilis ko magsalita. Francis Alipio, I haven't heard your voice for quite some time. What's the test for for motility? Nobody? No answer? What about Joanna Baltar? Natahimik sila nga, sir, eh. Medyo nag-alala ko ng a little bit. Alam ko medyo mabilis tayong magsalita, but because we need to finish this today. SIM, very good. So from time to time, class, I'll be calling your names. Uh, so please don't be scared. Please don't be intimidated. I just want to check if you guys are paying attention. Okay? From time to time, I will call you guys out. Because in my lectures, I know it's annoying. Some of you guys are being called. I know when I was a student, I know. Sir, ako na naman ang tinawag mo, sir, ganun, ganun. But please be mindful that whenever I call somebody's name, it's not because I want to embarrass you or make you feel unworthy of passing the exam. No, it means I just want to get your attention. All right? I know. Okay, so SIM. We use SIM. So for example, the organism grows in what? SSA. Okay. So we do TSI just to confirm it's actually TSI in the TSI and then IMVIC, and just to confirm whether or not the organism is Salmonella or Shigella. And then after that, we put we subculture it into a broth medium. So we subculture the organism into a broth medium. It could either be alkaline peptone water or what's the other one? Selenite broth. Okay, just in case we can't test it for now. And then we test for motility also. So IMVIC. So IMVIC is where we have indole test. So indole test will actually give us the result of what we're looking for. All right. Now, let's go on to Gartner's bacilli. Gartner's bacilli refers to what Ewing classification of Salmonella. It's actually Salmonella enteritidis derby, cervi tuloy. So, Ewing classification, what's the difference between them? There is only one difference. They added Salmonella enteritidis. All right? So Salmonella enteritidis. Zero var derby. Zero var tarpomerium. All right, so that's the only difference. Now we want to identify which one is Gardner's. We want to know who is Gardner's bacilli in the Ewing classification. Then that's the time you're going to say Salmonella, Enteritidis, Zero, Derby. Okay, that's the only difference. Now let's go on to Salmonella species that are usually the cause of bacteremia. We have Salmonella cholera suis and Salmonella typhimurium. Um, Salmonella cholera suis is the one that causes Salmonella cholera suis is going to cause bacteremia. Salmonella typhimurium, or if you want to sound posh and use the Ewing classification, you can say Salmonella enteritidis sero typhimurium cest. The other one is cesd. All right, cool beans. I can move on. No questions. All right, let's go back. Let's continue on. Okay, those are the answers. All right. Now, serogroups of Salmonella associated with enteric fever. Enteric fever. All right. And which one is the most common? Enteric fever. As I mentioned before, enteric fever is a patient who is suffering from diarrhea, vomiting. What else? Diarrhea, vomiting, and pain in the abdomen with fever associated with which is basically going to start out as fever. There are four species that you need to remember: paratyphi, Salmonella paratyphi A, paratyphi B, Salmonella cholera suis, and then lastly is Salmonella typhi. All right? The most common cause, of course, is Salmonella typhi. All right? Salmonella typhi, the enteric fever that causes the enteric organism that causes typhoid fever. All right? Please don't forget the difference between typhoid and typhus. Okay? Typhoid. O, di ba? Ang ganda-ganda ng sulat ni Sir Manuel sa ano. All right? Now, I will give you guys I will give you guys a question in the chat box. Um, what is the causative agent of the following? A. Typhoid fever, fever. And the other one is B. Typhus fever. So please don't forget these two. Okay? Okay? So what is the causative agents of the following? Hoy, wala akong sinabing A or B. Sinabi ko sagutan nyo A and B. B. Sir Marco, inaantok na si ano. Inaantok na sinaantok na si Mayra. Mayra, wala pa akong hindi yan choices. Pinapa-identify ko sa iyo kung sino yung causative agent. Nakakaloka ka. Nakakaloka. Bakit ka, bakit kayo ganyan? All right, I will leave that for you guys because we have to move on. There's a lot of material to cover. All right. So other names for other terms of Salmonella paratyphi B and paratyphi C. Oh my gosh, sir. Ang dami ko na naman kailangan alalahanin. So paratyphi B is Salmonella Schrottmülleri. Okay? And then paratyphi C is Salmonella Hirschfeldi. Okay? Paratyphi B, Salmonella Schrottmülleri. And then paratyphi C is Hirschfeldi. Alright? I know there's a lot of things to remember. Pero lagay lang ng lagay and sabi nga nila sa Board Exam Review Center, lagay lang ng lagay anyway, hindi naman ako magre-review niyan. Okay? So other terms associated with enteric fever, we also have typhus fever. Okay? Typhoid fever, sorry. Enteric fever is known as typhoid fever, not typhus ha. And what is the most common clinical feature of enteric fever? Well, you're going to see, of course, fever. Okay? And then you're going to see rose spots in the abdomen. Basically, what happens is that there is somewhat of petechia. It gets larger and larger the more severe the infection is. So basically, what this is what happens. For example, that is your patient, and that's the abdomen, belly button. Hindi tayo maglalagay ng boobs ha. So upon inspection, you will see rose spots over there. Okay? O, di ba? Very, very anime yung drawing ko, no? Rose spots. Ang anime ng itsura, no? Sir Marco, it looks like Eula. Charing. Charot. Charot lang. All right. What is the preferred sample for the diagnosis of Salmonella? Actually, in microbiology, I need you guys to remember Salmonella, or specifically typhoid fever, is the one of the diseases that needs to have a specifically timed specimen. 1 week after the onset of signs and symptoms, you have to collect the blood, test the patient for what? For serological testing. Test the blood rather for um, blood cultures. For the second week of the onset of signs and symptoms, you have to test the patient's stool, and on the third week, you have to test the blood again. But this time, you have to test the serum. You need to subject the patient to serological testing. And what is the serological test for typhoid fever? The serolo that is the question. You have Widal test, the Widal test. Okay? What are the antigens present in Salmonella? There are again antigens. Ayan na naman yung antigens, sir. Nakalimutan ko na. Ano nga yung antigens? What are the antigens? Ayan, gising sila. Lovely. Gusto ko gising kayo kasi medyo mahaba-haba pa to. Ilang slides pa ba to? Nakakaloka. 260 na sa 150 pa lang tayo. All right. So you have to remember these antigens. Okay? Please, I've discussed this earlier. OHK, somatic, tandaan may O siya. Silang dalawa wala. K, capsular. H, bahala na kung paano nila pinangalanan, basta flagellar yan. Alright? Walang kakwenta-kwenta pero yun yung naalala ko. Alright? Next, let's go on with the differentiation of O and H based on stability. Again, this is something that you need to remember again. O antigen is heat stable. Why? Because what? Because it's larger. Of course, the protein strands are also complex, are much more complex than the straight line, than the straight or rather single filed protein structure of your flagella. Okay. Now the VI antigen is known as an antigenic structure, is known as the capsular antigen. All right, it's the capsular antigen. So aside from knowing it as K antigen, it is also known as the VI antigen for this particular organism. K antigen is for most organisms, but for Salmonella, it's VI antigen. All right, and this antigen is actually responsible for the invasiveness of the organism. Okay. All right, so indicate the titer values of patients with the following typhoid conditions. Active infections, they will have greater than 1 is to 60. Okay, 1 is to 160 rather. 1 is to 160. For immunized patients, for 1 is to 1 is to, sorry, for this is for the O antigen. Okay, so for immunized patients, usually what they're testing is the H antigen. But you can test for the O antigen as well. But I doubt there will be a reaction. Okay, so for immunized patients or immune patients, they will have greater than 1 is to 60 in the H antigen. H antigen. Okay, for carrier state, these are patients who can transfer the stool tuloy. These are the patients who can these are the patients who will these are the patients who can carry the organism around and can basically basically cause a community infection, or um, an endemic, no, not an endemic, I forgot the terminology, we'll come back to that one later. But the carrier state will test positive or greater than 1 is to 60 for the VI antigen or your K antigen. Okay. All right. So how many samples are obtained for Widal tests? There's two samples. One one sample is collected in a 7 to 10 day interval. All right. One sample every 7 to 10 days. 7 to 10 days. All right. Reason is to prove there's an actual rise in the antibody titers. Okay. So we want to check if there is any changes in the patient's titers. Okay. Time interval for each collection. 7 to 10 days interval. There are two samples that we collect. One during the patient's admission, and then the second one in a 7 to 10 day interval to prove the actual rise in antibody titers, because this is a test that is not normally done in routine analysis. Titers are usually done on serologic tests for hepatitis, what else? Hepatitis B, hepatitis C, but rarely done on bacteria. So in this case, usually for Widal tests, we want to collect one sample upon admission or several hours upon admission, and then another sample after treatment. After treatment or slash diagnosis, and then we want to uh, um, the need for this is because we want to see if there is actual rises or falls in the patient's results. All right, so black colonies is observed in what culture media? Black colonies of Salmonella typhi. Black colonies of Salmonella typhi is observed in bismuth sulfide agar and xylose dextrose agar. Okay? And bismuth sulfide agar is actually the best culture media for demonstrating the black colonies. There's supposed to be a picture here, but I think because this is the unauthorized version, the uh, update actually this deleted it by I don't know what happened to it, but there was supposed to be a picture of it here. Anyway, you can just proceed and do a Google search of Salmonella typhi in bismuth sulfide agar, and that's how basically I did it anyway. Now let's go on with the IMVIC results of the following. Salmonella typhi will test positive with only methyl red and negative for the rest. Okay, but other salmonella will have the following results. Okay, oops, typographic error. Wait, let me end the slideshow for a while. Score supposed to be negative. All right, so this is the results of the other salmonella species. Okay, salmonella typhi will have negative, positive, negative, negative. IMVIC, we're talking about IMVIC here. Other salmonella will test positive, negative, positive, negative in IMVIC. See the results again of Vogue's Proscour and methyl red. They're always opposite. See? See the difference? Now, let's talk about Salmonella type, other name for Salmonella type B. This is also known as the Edwards bacilli. So Edwards bacilli is Salmonella typhi. All right. Now we're going to move on with Shigella. All right. Shigella is the natural inhabitant. The natural habitat of Shigella species is limited to what part of the human body? It is the intestinal tract. It cannot survive in the other places in other places around the human body, and you'll know later why. All right, there are four groups of Shigella species that you need to identify, that you need to be familiar with. It's basically group A to D, but each letter is different. Each letter has a nomenclature nomenclature wait. Saan na ba ako? So this is the one, but I need you guys, I need to correct something. Uh, ay, sige. Oh, yes. Oh, all right. Medyo nag-commercial si sino ba 'yon? Medyo nag-commercial si Arisgado Lindel. But anyway, this is the groups and this is the complete nomenclature of the organisms. All right? Group A, Shigella dysenteriae. Group B, Flexneri. Group C, Boydii. Group D, Shigella sunnae. All right? And these are the tests on how to differentiate them. Okay? Everybody will test positive for mannitol fermentation. So please be mindful of mannitol fermentation. All right? Ay, laser pointer na para cute. All right, mannitol fermentation will be positive for all of them. And then ONPG will identify Sonnet as a late fermenter. All right. And then catalase will be negative for Shigella dysenteriae. Is it important for you guys to remember? Yes. Everything I put here has appeared once or twice in the board exam, and it doesn't appear like this, guys. It will not appear like this. It will appear in a descriptive sentence. It will appear in a descriptive sentence, and you have to you figure out how to answer it. If you don't have any idea on how you will be able to, if you don't have any idea about this particular chart, then you will not be able to answer that question. So please compartmentalize what you need to remember. Mannitol fermentation, I know everybody will test positive for mannitol fermentation. ONPG, I know everybody will test negative except for Shigella sonnae, which is a late fermenter. I know everybody will test positive for catalase except for Shigella dysenteriae. So please compartmentalize what you need to learn because this is going to be important in the exams. All right, now let's move on. Um, Let's move on. What are the toxins elaborated by this secreted na lang? Ano ba yung elaborated na yan? Masyadong luma tong mga terminologies na ginagamit ko. Palitan natin. Discard. Secreted na lang. Oh, ayan. Para, bakit ba elaborated? Yung professor ko kasi naglagay niya ng question na yan. So, ginaya ko lang dati. Alam mo naman, bata, innocent pa, naive cells, ganon. All right, so what are the toxins secreted by Shigella dysenteriae? So you have an exotoxin and an endotoxin. So you have an exotoxin and an endotoxin. There are functions of each, and the exotoxin is responsible for um, is the toxin that is released during intestinal colonization. Remember that as I mentioned before in our discussion with E. coli, the difference between Shigella and the other Enterobacteriaceae is that Shigella is invasive. And one ways we can identify it. One way scientists were able to identify it as a colonizer or as an invader is through the secretion of an exotoxin. And endotoxins, basically what happens is when the bacterial cells break up, this will result in bacterial irritation. All right, it's because the antigenic structures resemble that of um, chemical biochemical messengers that have certain effects on the uptake and the reabsorption of water in the large intestine. All right. So the appropriate specimen for Shigella is two things. You can either use stool for culture or you can use rectal swabs. Now, how do we process rectal swabs? You process the rectal swab as a stool sample. All right. You process the rectal swab as a stool sample. Okay. Regardless of whether it's bloody or not, you have to test it like that. Sometimes kasi guys, in the laboratory you will get perianal swabs and it will come from hemorrhoid origin, so pustular hemorrhoid origin, or it can come from a wound abscess in the perianal canal. Okay, so rectal swabs in this with the indication of enteric fever, question mark enteric fever in the diagnosis, you need to process it like a stool sample. It's not like a wound swab. So what's the difference between the processing of stool samples and other purulent swabs? When I say purulent swabs, there's pus in it. I'll give you guys a breakdown of how they are processed. Okay, in most laboratories that have been accredited by American accreditors, they will have this. Say for example, this is stool, this is pus samples. Okay, all right. So stools are usually inoculated in two things: broth media and solid media. Solid media includes your SSA and McConkey or sorbitol McConkey, smack na lang. Okay? And the broth media would include what? It would include selenite broth and what else? That's the other one? Alkaline peptone water. I forgot to mention you have to test also. Usually when you're doing stool analysis, you have a stool culture. You have to also add you also have to add H. pylori medium H. pylori medium, and Campylobacter medium, Campi agar, and H. pylori medium. Both of these, the only difference of this is that these are blood agar plates with antibiotics, but and they are also introduced in a not CO2, microaerophilic packets. You need to use microaerophilic packets. So that's the breakdown of how stool samples are analyzed in the microbiology laboratory following College of American Pathologists protocols or the CLSI protocols. Now for pus samples, it's different. It's an entirely different game plan. How we do this is you incubate the patient in another again in broth samples and in solid medium. So for broth sample, you need to have thioglycolate broth or TG broth, thioglycolate, and then for solid medium or agar plates, you need to inoculate it in blood agar, chocolate agar, McConkey, and then and then lastly is anaerobic blood agar. All right, anaerobic blood agar. You get the difference why we need to process it as a stool sample. All right, rectal swabs are processed as stool samples following the CLSI protocols. Okay, because pus samples target pus related organisms. Okay, whilst this one will target or will selectively grow enteric pathogens. When I say enteric pathogens, these are organisms that are usually pathologic to patients, usually pathologic to the intestinal tract. All right, clear? Clear ba? Is everything clear? Clear? Clear ba, Adrian? Clear ba, Joanna? All right, clear. Okay, so you got my idea. This is not part of your lecture because this is actually clinical practice. So I just want you guys to have a little bit of an understanding on what's the difference between processing a stool sample and processing some pus samples. Okay, so it's not part of that one, and I don't think it will be part of your board exam because this is more on the clinical portion. I just want to give you guys an idea. All right, so Shigella dysenteriae is very strange. I've mentioned before that all the tests for Shigella will test negative for a lot of for most of the biochemical tests. But let's go back. Um, as I mentioned before, Vogue's Pro Scour and Methyl Red will have opposite results again. So Imvic, Methyl Red, and will have opposite results. And the funny thing about Shigella dysentery is that it will produce a variable results in indole testing. All right. Now, let's go on to the other names. So, nomenclatures. Shigella dysenteriae is known as the Shiga bacilli. Flexneri is known as the strong bacilli. Boydii is Boyd's bacilli. And Sunei is Sonduval bacilli. Easy to remember. Shiga bacilli, dysenteriae. Flexneri, mahilig nga siyang mag-flex, di ba? Kaya strong siya. Boyd's, from the name itself, Boyd's, Sunei, Sonduval. All right, easy to remember, clear? I don't hear anything. All right, wonderful. Now let's move on to the classification of Shigella species based on what particular, the classification rather of Shigella species are based on what particular antigen. It is the somatic antigen. Each species of each species that we're talking about here has a different structure of their O antigen or their somatic antigen. All right, and what antigen is present only in the Shiga bacilli? It is actually the A antigen. Shiga bacilli. Another name for Shiga bacilli is your Shigella dysenteriae. All right, so clear? All right, now we can move on to the next organism, Proteus species. Proteus species. Remember that when we're discussing Proteus species, as I mentioned before, you need to remember the other organisms that are supposed to be discussed with them. What are these? PPM. Usually it's compartmentalized together in a lot of the discussions. So please don't forget PPM. What other organisms do we need to discuss aside from Proteus? Because they have almost similar characteristics. Anybody? Oh, magtatawag na naman ako because they're very quiet. And all right, wonderful. All right, wonderful. Who was that? I didn't see the name. Ah, it's Joanna. Thank you so much, Joanna. All right. Now, let's talk about the characteristic species that produce a translucent sheath in the culture media. The characteristic of Proteus species to produce a translucent sheath in the culture media is what you call the swarming phenomenon. So I'll describe swarming phenomenon for you guys who have not participated in their lab experiments in the hospital in the laboratory. So basically what you do is you inoculate it 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, right? So you didn't inoculate here, you didn't inoculate in this part as well. But because there is what you call swarming phenomenon by sorry, ayusin ko yung drawing ko. Sabi kasi ng friend ko na nanonood sa akin, nag-message siya sa akin, hindi niya maintindihan yung drawing ko. Nanonood kasi siya sa via YouTube, nag-message siya sa akin. So for example, na-conscious tuloy ako. All right? So what she said is that I need to draw properly. So inoculate here, here 1, 2, 3, 1, 2, 3, and then 1, 2, 3, 2, 3, and then 1, 2, 3. So this is going to be in a circular plate. So I did inoculate at this part, right? I did inoculate at this part, and I didn't inoculate at this part. But after 24 hours of incubation, if the organism is Proteus, it will grow, it will make a translucent sheet. It will grow colonies here, huh? But it will also grow, it will also leave a translucent sheath of growth. And it smells extremely foul for me. And I hate with I hate it when I encounter this organism because it can cause mixed field culture. All right, so that's a swarming phenomenon. Basically what happens is that it swarms the agar. All right, so what substances are incorporated in the culture media to prevent swarming phenomenon? So you can use sodium azide, you can use phenylethyl alcohol, and chloral hydrate. Look at these things. These are some things, see, these are reagents that inactivate the flagella, okay? It inactivates the flagella. So it inactivates the organism's flagella so as to prevent the swarming phenomenon. Why? Because it is, as I mentioned before, Proteus species are notoriously difficult to set aside to grow alongside other organisms. And it is notorious, especially if there are two organisms growing in the culture media. So you have to subculture the whole thing or the whole specimen in a media that contains the following, any of the following. And in the most common media that we use is Kled. This is actually used for urine samples, but for practical purposes, it has sodium azide, which prevents the swarming of the colonies, and it's been very helpful for me when I was practicing, when I'm doing urine cultures. All right, now differentiate the strains of this Proteus species inoculated in culture media. When different strains of Proteus species are inoculated in a culture media, a demarcation line appears in the swarming in the swar in on swarming species. This phenomenon is known as the Dane's phenomenon. So what is Dane's phenomenon ba? So for example, this is your culture media. Some hospitals they separate it just to save the culture plates, so that's one thing that you need to remember. Okay? So imagine that is the case. So one past sample was inoculated here. One, two, three, one, two, three, and then like that. And then another one here. One, two, three, one, two, three, one, two, three, like that. All right. So supposed to be there, if it's Proteus, imagine as you imagine. I assume that these two isolates are pure isolates of Proteus. And what happens is that they are both genetically different strains. And what they do is that they won't mix with each other. They will form a demarcation line when they swarm. A demarcation line basically separates both ones. So imagine you just need to imagine both of them fighting over one agar plate. Okay? So that's what you call Dane's phenomenon. There's a demarcation line that separates two different strains of swarming Proteus species. All right. So what particular antigen is used for serological testing? We again use the O antigen, and and we use this O antigen in what you call the whale Felix test, not Widal test, for whale Felix test, and this is useful for the diagnosis of rickettsia. Okay, rickettsial infection. Okay, whale Felix test, O antigen from Proteus species. Okay, useful for the diagnosis of rickettsia. So that's just added tidbit of information. Now let's move on to antigens used in the whale Felix test. So you use the OX and the OX19, OX2 and OX19 Proteus vulgaris antigens, and then for Proteus Proteus mirabilis, you use OXK. All right. So if you encounter these terminologies in your serology, know that this is used for whale Felix test, and know that these are the organism sources are Proteus species, specifically Proteus vulgaris and Proteus mirabilis. All right. Now, because Proteus species can produce an alkaline environment, they have been associated with what you call urinary calculi and urinary stones. Now, why is that? Because the normal urine is supposed to have an acidic to neutral environment. It's not supposed to have an alkaline environment for prolonged periods of time. So in cases of uh, in cases of urinary tract infections associated with Proteus species, you are going to experience these are patients who are going to experience urinary calculi. All right. Now, IMVIC results. IMVIC na naman, sir. Yes. wag niyong kakalimutan. Proteus vulgaris will have the similar reaction, a similar reaction to E. coli, but the last one, the citrate utilization test, is the different one. Proteus vulgaris will have a variable result, while Proteus mirabilis will have a variable result on Voges Proskauer and for citrate utilization. Clear? Everybody can still follow? All right. So everybody can still follow. So if everybody can still follow, we can move on. All right. Good. Wonderful. 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 So these are the species that are capable of swarming blood agar plates. This is Proteus mirabilis. All right. Usually Proteus species will be able to swarm only in McConkey agar and chocolate agar. But in blood agar plates, you can actually see Proteus mirabilis do this. All right. Now, how would the following Proteus species be identified? Supposed to be Proteus vulgaris dapat. How would the follow? I'm supposed to write here. I don't know why it disappeared, but I'll try to fix it for you guys. And slideshow. How would the following Proteus species be identified? Mm, Proteus vulgaris and Proteus mirabilis. So there's one test to differentiate both of these things, both of these organisms. It's actually indole test. All right. Look at the difference. See, Proteus mirabilis is positive, while Proteus vulgaris is positive, while Proteus mirabilis is positive for I, which is indole test. Let's see the difference. Proteus vulgaris will test, Proteus mirabilis will test negative. Proteus vulgaris will test positive. Look, double check if my statements are correct. They are correct. All right. Can we move on? All right. So next, production of gas. Let's talk about the production of gas. The production of what gas is the reason why protein species can be identified from the other members of Enterobacteriaceae. It is capable of producing hydrogen sulfide gas. All right? It is capable of producing hydrogen sulfide gas. And what test is used to differentiate? What test is used to differentiate or identify the gas-producing organisms, the gas-producing species of Proteus? Well, we have hydrogen sulfide test. And another thing that you can use is phenylalanine deamination and urease test. Again, review. Where do you do phenylalanine deamination? Phenylalanine deamination. You use it in lysine iron agar. Okay? And then urea test, you use it in the urea agar or urease agar. And what reagent do we add in the urea agar or the urease agar? What reagent? Kovacs or Ehrlich reagent. All right, you use Kovacs or Ehrlich reagent. What is the content that is important or the chemical that is important in both of this? Padma. Okay? I know ang pangit ng sulat, no? I know my handwriting is iffy. This is computer. I don't have a pen. All right? So that is the organism that we're going to talk about. Oh my gosh, we only have 60 slides to talk about. 60 slides. We're at number 206. We only have 54 slides to look at. All right. Now, we're going to move on with Citrobacter and Arizona species. Now, Citrobacter and Arizona species are closely related to the Salmonella species. Okay? They are closely related to Salmonella, and they are morphologically similar, or rather, um, let's talk about the similarities. Citrobacter is morphologically similar to E. coli, but they are biochemically similar to Salmonella. So this one is they are close relatives of Salmonella species on a genetic level. But And then if we're going to look at it in a much more simpler way, similar morphology wise, E. coli. Biochemical, from a biochemical perspective, they are similar to Salmonella typhi. All right. Now, what tests may be used to differentiate Salmonella from Citrobacter? Well, again, we have to use urease test, and actually, Citrobacter can grow in KCN agar. All right, KCN agar. What is KCN agar? Potassium cyanide agar. It can grow in potassium cyanide, unlike Salmonella. All right, clear? You have to use urease test, and you must demonstrate its growth in KCN. Now, IMVIC results. IMVIC results is positive, negative, positive, negative. Huh? Please don't forget. Positive, negative, positive, negative. Now let's talk about Arizona Hinshawi. Arizona Hinshawi is an accidental pathogen to man. What animals are usually infected by this bacteria? It's actually birds and reptiles. All right. So you might get a case presentation of wound swab collected from a zookeeper who usually works with crocodiles or any reptile or snakes. All right, or a veterinarian was bit by blah blah blah, and pus formation happened, something like that. You might get case presentations like that. Okay, and then they will give you the presentation. They will give you that it grows in Salmonella Shigella agar. Upon testing, it tested urease positive, and it was able to grow in potassium cyanide agar. Then that's the time you're going to select Arizona species. All right. Now, utilization of what sugar is unique to Arizona Hinshawi. All right. One way to differentiate Citrobacter from Arizona is that it is capable of utilizing malonate. Okay. So malonate utilization results to an alkaline pH in the environment. The color indicator is bromthymol blue, and the positive result in the utilization is again color blue. All right, remember, if bromthymol blue is the indicator, the positive result in an alkaline environment, the positive result um, for an alkaline, the positive result is an alkaline environment, which is characterized by having a blue color. All right, if it becomes acidic, it will turn yellow. All right. So again, Adrian, what is the color of your Simon citrate agar without doing anything to it? Tulog na. Patay tayo. O sige, Joanna, answer for me. All right, wonderful. All right, let's move on to the imbic reaction of Arizona Hinshawi. So it will have an opposite reaction to that of Citrobacter. It will be negative, positive, negative, positive. All right. Now let's talk about species closely related to these things. We have Edwardsiella. So Edwardsiella is going to infect the following organisms, or can be isolated in the following animals: reptiles, freshwater fish, aquarium fish, turtles, and frogs. So again, you have to be familiar with where they're taken from, because they might present you case studies and give you IMVIC results of the following organism of different organisms, and that's how you can differentiate them. All right. Now, the species of Edwardsiella usually isolated in man is Edwardsiella tarda. All right. This infection, these conditions have the following effects. You can have wound infections, septicemia, and meningitis. All right. Remember, wound infections. patient was a zookeeper, or patient was just preparing, or the patient was just cleaning the aquarium, or got an infection from broken aquarium glass. Oh, you already have your clues there. And then they will give you in-vic reactions similar to that one. All right, so Providencia and Morganella. We're gonna go with Providencia and Morganella. Okay, Providencia and Morganella are closely related to what organism again? Mera? Proteus. Okay, sinabi mo na Proteus, nagsorry ka pa. Ikaw talaga. All right, so both organisms can be differentiated from Proteus because they do not produce hydrogen sulfide. All right, they cannot produce hydrogen sulfide. And Proteus is notoriously known for producing black colonies in your TSI. All right, so medical conditions associated to Providencia retgeri, it has been associated with Providencia has been associated with UTIs, and Morganella morgani is a common nosocomial infection. All right, next, in-vic results. Dinagdagan ko na naman kasi gusto ko kayong mahirapan. All right, so Providencia retgeri and Providencia stewarti will have the same in-vic results as that of uh, yeah, so we'll have the same in-vic results of as each other: positive, negative, positive, 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 negative, positive, 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 negative, positive. Whilst Morganella morgani will have the same reaction as that of what organism? E.coli na naman. Okay. So next, we're going to talk about Yersinia. So Yersinia is also known as your plague bacillus. All right. And the characteristic stain using Wason stain is what you call the characteristic morphology when stained with Wason stain is the safety pin bacillus. What do I mean by safety pin bacillus? It will look like this. Okay, so it will look like that. There's a polar swelling, I suppose. Now, what antigens allowed Yersinia pestis to be more susceptible to intracellular killing, to be less susceptible? Sorry. So we have the the W and the V antigen. And there are several toxins that are produced by there is a toxin produced by Yersinia pestis. Please don't forget that it is an exotoxin, and this toxin is known as pesticin or bacteriocin. Bacteriocin. All right, so pesticin or bacteriocin. That is the exotoxin produced by Yersinia pestis. Okay? Now, enzymes elaborated na naman. Released na lang nga. Bakit ba kasi mahilig yung professor ko na magsabi ng elaborated? Nako-confuse tuloy yung mga estudyante. Released na lang ha. Please change that to release. Enzymes released by Yersinia pestis, we have coagulase and isocitrate lyase. All right, so these are the enzymes that are present in your plague bacillus. Now let's talk about the plague. Plague has three types. You have bubonic plague, you have pneumonic plague, and you have septicemic plague. So bubonic plague is characterized by just a couple of lumps, dark to bluish to dark black lumps in the lymph nodes, while the pneumonic plague is the continuation of the bubonic plague. So basically, this is level 2, and then lastly is level 3. So aside from having bubonic lumps in the armpits or anywhere where there's lymph nodes that can be palpated, pneumonic plague has the addition of respiratory syndrome. So we have hemoptysis, cough, and everything like that. Now, as it goes into the respiratory system, it will make its way to the circulatory system and become septicemic plague. And it has been actually the cause of the Dark Ages. This is one of the reasons why in Europe it was the Dark Age. This was considered as a Dark Age. The why it became a Dark Age in Europe. It's killed millions of people actually. All right. Now, what is the mode of transmission? Oh my gosh. This is a question for PAMET, for the PAMET quiz show. Okay, when I was a student, I remember this so much. So I want you guys to familiarize yourself with the mode of transmission of Yersinia pestis because it is through a vector bite. But what particular vector? The flea, uh, tick flea, what you call this? Fleas. It comes from fleas, from rat fleas, particularly known as Xenopsilia chyopis. All right. So a vector bite from Xenopsilia chyopis, which is basically rat fleas. Okay. So traditionally, they thought it was the plague was caused by respiratory droplets, and so they wore masks like this. I don't know if you're familiar with the medicine man in Europe, but this is what it looks like. There is a bunch of antibacterial herbs inside, or herbs rather. There's herbs inside because they thought it was from because at that time they thought it was from inhaling what you call miasma. What is a miasma? It's a noxious gas that can cause disease, something like that. But they didn't know that it was from Xenopsilia chyopis. And after this discovery, they realized that it was a community-acquired infection from rats because in the Middle Ages, they didn't have any idea about public health and hygiene, so rats were running rampantly in different places around Europe and even in the early American colonies. So plague was such a devastating blow to the country. Now, black death is a term associated to the plague bacillus characterized by what clinical picture? Black purpuric spots, of course, and the term for this one is what you call the Schwarzmann's phenomenon. All right? So black purpuric spots. So it's known as the Schwarzmann's phenomenon. Okay, and some cases it sometimes appears in places around the world, specifically in European countries where there's not enough hygiene or water. Okay, now let's talk about broth cultures. In broth cultures they have what you call stalactite formation. All right, so if you have thioglycolate broths and you incubated Yersinia pestis there, it will form stalactite formation or stalactite streamers. So basically what happens is that this is your test tube, and then that's your broth media. Of course, this is capped. There's a cap on the top. And it will form stalactite formation or stalactite streamers. Why did I call it streamers? Because when you move the agar around, it looks like, when you move the broth media around, it looks like a couple of streamers that you put on your part in parties as decoration. Now, red-colored colonies of Yersinia pestis is seen in desoxycolate agar or deoxycolate agar, sorry, not desoxycolate, deoxycolate or the DCA agar. All right, and the hemolytic pattern of um, of Yersinia pestis in blood agar plates is that it's gamma hemolytic, of course, as opposed to the many members of Enterobacteriaceae that we mentioned earlier. Most of them are alpha hemolytic, are gamma or are beta or gamma hemolytic. But in this case, Yersinia pestis is more of a gamma hemolytic organism. All right. So this is a zoonotic species of Yersinia that has been isolated upon ingestion of unpasteurized milk. It's Yersinia enterocolitica. All right. It causes enterocolitis. Okay. And usually we can isolate them in the CIN agar, Cephsulodin irgasan novabiocin agar. And it has what you call the bullseye appearance or bullseye colonies, Cephsulodin irgasan novabiocin agar. And I think that's the end of our discussion for Enterobacteriaceae. I'm gonna give you guys a couple of minutes of break, just to gather your thoughts, I suppose. And I think that's it for today's lecture. I think that's it. But there's going to be a break because I'm going to give you guys an activity. Or would you rather we continue on with gram-positive bacteria? I'm going to give you guys a decision because Sir Marco is the one who is going to give you guys the exam. So I want you guys to be exposed to as much material as you can. If it was me who was making the exams, I would make it difficult, but I would only ask questions related to the topics that I've discussed. But since Sir Marco is the one who's going to make the exams, you need to be exposed to as many materials as you can. So I'm going to ask the class, should we continue with a different with the next topic, or should we just remain here and continue on tomorrow? Ay, walang gustong sumagot. Oh, walang gustong sumagot. Ay, wala. Oh. So paano guys? You guys decide because you have a couple of minutes left and I think there's time for us to discuss the other gram-negative organisms lecture. But whilst you decide, whilst you decide, I'm going to give you guys 5 minutes to decide because I'm not the one who's going to make the exam, guys. Remember, it's Sir Marco. So if you're not exposed to as many materials or content as you can, you may not be able to answer all of his questions. So think think clearly and decide amongst yourselves. Would you rather? Uh, so decide amongst yourselves because we only have one more lecture for tomorrow, and I'm going to dedicate that for gram-positive organisms. So I'm going to give you guys 5 minutes to decide whilst I go and prepare my lunch.